Hello and welcome to our third video for chapter 21. We are going to start with dynamics of blood circulation. So the first thing that we have to mention is that the movement of blood within our vessels are going to be based on three major topics, which include pressure, flow, and resistance. So we're going to focus a little bit more on the flow right now. You could see that I have pictured here for you a laminar flow and a turbulent flow. When we have laminar flow, you want to imagine blood moving through a vessel more in concentric circles, kind of the way it's illustrated here. And what we want to realize is that the blood that we find closer to the blood vessel wall is going to experience greater resistance to flow. This is why the arrow is a bit shorter than compared to blood that we find closer to the innermost layer that will just slip over the other concentric layers back in here. And that's why we see a longer arrow here, because they will experience less resistance. You can compare this to the flow of water through a river. Now, if we have a rock or a more narrow region in that river, we are going to see that there is turbulent flow. So basically, turbulent flow is any interruption that we have within laminar flow. This can happen when the flow is going to exceed a critical velocity, or when the fluid becomes constricted, like we said, if the river becomes more narrow, or there's a sharp turn, maybe there's a rock over here, or if we're talking about the body, it would be a thrombus, or some type of rough surface if we have atherosclerosis. And when we have this turbulent flow, typically we are going to hear sounds because of it. So we can hear sounds as blood moves past valves in the heart or in areas where the artery is going to branch. These would be normal sounds that we expect to hear. But if we hear a sound where we should not be hearing a sound, it's probably because there's some type of abnormal constriction within the blood vessel, and we can expect for there to be some type of thrombus there. To define what blood pressure is, we can say that it's a measure of force exerted by blood against the wall. In order for blood to move through the blood vessels, we need pressure. And we can measure this by listening to the Korsakoff sounds produced by turbulent flow in our arteries as pressure released from the blood pressure cuff. So you may be familiar with taking blood pressure already. If you want to get a more direct reading, then you would want to place a catheter within your patient in a blood vessel, which could be like the femoral artery, for example. Or you can also place a catheter into a chamber of the heart to monitor any pressure changes. But this typically is not used when a patient comes in just for a clinical exam. So health professionals are going to use a oscillatory method. This is going to involve wrapping a blood pressure cuff, which you see pictured here, connected to a sphygmomanometer, which we are using this illustrated portion here. And we place a stethoscope over the brachial artery. Once we have everything in place, we are going to create pressure within the cuff that is high enough to keep the brachial artery closed. So you can see at this point, the cuff is inflated and the brachial artery is collapsed and closed, and you will hear no sound at this point. Then you start to decrease the pressure in the cuff, which keeps the brachial artery closed, but blood is going to start to partially move through as the brachial artery begins to open a little bit. So once again, we have a turbulent blood flow that's going to create a sound for us. And that sound is going to be measured as the systolic pressure. The cuff will then continue to deflate, which opens up the brachial artery more and more until we are going to stop hearing sounds, and that we will record as our diastolic pressure. At this point, the blood flow turns into more of a laminar flow, and that's why we don't hear any more sounds. By using this method, we are at least within 10% of methods that are more direct. When it comes to resistance or peripheral resistance, we are discussing opposition to flow. And we can measure this as the amount of friction blood encounters with our vessel walls are generally in our peripheral or systemic circulation. And this is going to be based on three important sources of resistance. First, we have blood viscosity, which is the thickness or the stickiness of blood, and we'll discuss some details as to how this affects blood pressure and flow. We also can consider total blood vessel length. So the longer the vessel is, the greater resistance we will have. And blood vessel diameter. So if the radius increases, like we see in vasodilation, then resistance is going to decrease and vice versa. We can use a calculation in order to get our peripheral resistance that will consider all of these factors. But I'm not holding you responsible for memorizing that equation, and I will not have you do any math on your exam. So if we just take a look to the right here, we could see that when we have low diastolic pressure, it's due to vasodilation of our arterioles, which is going to allow low peripheral resistance. And when we have a high diastolic pressure, that means that we have vasoconstriction going on, and that gives us high peripheral resistance. Now a little bit more on viscosity. Viscosity is a measure of resistance of liquid to flow. So as viscosity of our blood increases, then the pressure that we need in order to push that blood through the blood vessel is also going to increase. So if we have something that's highly viscous, like syrup, we would need a great amount of pressure in order to push that through a tube. We're going to see that viscosity is largely influenced by hematocrit, which, if you remember from chapter 19, is the percentage of the total blood volume that's composed of red blood cells. As our hematocrit increases, our viscosity is going to increase as well. And anytime we're dehydrated or have a uncontrollable production of our red blood cells, that can lead to an increase in our viscosity from being at the normal level of about 45% to, let's say, for example, 60%. And that means we have the viscosity of about 6 times more of that compared to water, which is going to increase the workload on our heart, and that can lead to heart failure. Vascular compliance is the tendency for your blood vessel volume to increase as blood pressure increases. I like to think of this as just compliance in general, so that as we have a blood vessel that likes to stretch as pressure increases, that means that it has a greater amount of compliance. So let's compare this as if our arteries were a plastic cup and our veins were a water balloon. So if we apply more pressure to that plastic cup, we are not going to be able to hold more volume of blood in there just because we increase the pressure. So if you look at the figure here on the right, you can see that intravascular pressure is increasing, but the arteries are not able to hold as much volume with that increase in pressure. Whereas with our veins, when we fill that water balloon up with a greater volume, it is going to be able to adapt and increase its volume as the pressure increases. Our venous system is going to have a large compliance and allows it to be 24 times greater than that of arteries. And this really is because that they have thinner walls than arteries, so they're able to kind of hold more of the blood and serve as a blood storage or reservoir. About 84% of our total blood volume is found within our blood vessels. So about 64% of that are found within our veins, and 15% of that are found in our arteries, and 5% is found within our capillaries. Now let's discuss the physiology of our systemic circulation. This is going to be determined by the anatomy, the dynamics of blood flow, the regulation mechanisms that are going to control the heart and blood vessels, And we've already discussed how our blood volume is contained mostly within our veins because they have greater compliance, and the smaller volumes are found within our arteries and capillaries. When we look at the cross-sectional area of arteries versus capillaries versus veins, we're going to see that as the diameter of a vessel decreases, the total cross-sectional area increases and the velocity of blood flow is going to decrease. This is especially apparent when we look at the capillaries here. The diameters have decreased at this point, and we have a large cross-sectional area, but the velocity of this blood flow is going to decrease. We want this to take place because we want exchanges between the tissues. We want to get that oxygen off to the tissues, pick up the carbon dioxide or any other waste products, and then send that blood off on its merry way. Kind of like a stream that is going to flow rapidly through a narrow gorge, like the aorta or the arteries, but it's going to slow down through a broad plane. When we compare the different blood pressures within our blood vessels, we're going to see that we have the highest amount of pressure within the aorta. That averages at about 100 millimeters mercury. That is then going to drop to about 0 millimeters mercury by the time the blood gets through the arteries, the arterioles, capillaries, venules, veins, and the vena cava, and back to the right atrium. This is because we have decreased resistance to flow as the cross-sectional area increases. So you can see that we have a lot more fluctuation here between the systole and diastole pressures. But once we get to the small arteries and arterioles, we do not have those fluctuations take place anymore. Therefore, we say that our arterioles are going to regulate blood flow through our tissues. The term pulse pressure is going to be the difference between our systolic and our diastolic pressures. You may be familiar that our blood pressure normal reading is around 120/80. So if that was the case, we would take 120 and subtract it from 80 in order to get our pulse pressure. In the example down below, we have a systolic pressure of 110 and diastolic pressure of 68. We subtract them and we get the pulse pressure of 42. We'll see that this pulse pressure will also increase when stroke volume is going to increase. So for example, when you are exercising, that causes an increase in stroke volume, or it could be due to vascular compliance decreases. As you age, that typically happens, and the pressure is going to rise. Our pulse pressure can be used in order to take a pulse and determine the heart rate and rhythmicity. So we're going to look at a couple of the more frequent sites to measure pulse, but the most popular one is going to be at the radial artery. So you would find the radial artery right below the area of the thenar muscle group, that bulk at the bottom of your thumb, just proximal to that in the wrist region or the carpal region. Other areas we can take a pulse is the brachial artery, and this would be the medial side of your arm. You can also take the axillary artery pulse. You can use your facial artery, the superficial temporal artery, the common carotid artery is another very popular area to take the pulse, or the femoral artery, which you already know you can find in your femoral triangle. We can use the popliteal artery behind the knee, the dorsalis pedis artery, that would be anterior to our ankle, and the posterior tibial artery that you would find just posterior to that medial malleolus. Next, let's discuss capillary exchange and how we regulate interstitial fluid volume. So we've already discussed how the capillaries are going to exchange substances with the tissues, and the most important means of exchange is going to be diffusion. When we have a lipid-soluble molecule that crosses our capillary walls, it's going to diffuse through the plasma membrane. So examples of this would be like oxygen or carbon dioxide, steroid hormones, or fatty acids. But when we have water-soluble molecules diffusing, it has to be through intercellular spaces or through fenestrations of capillaries. or we can also sometimes use certain transporters to get amino acids through the cell and into our capillary. Another example would be glucose. Things that could affect movement of molecules through the capillaries is blood pressure, the capillary's permeability, depending on which type of capillary we have, and osmosis. What we typically see is that fluid will move out of the capillaries at the arterial end and into the tissues. And as we move closer to the venule end, we'll start to see that all of that fluid that we moved out at the arterial end is being placed back into the venous end of the capillaries, but about 10% will remain in the tissues, and that'll be picked up by the lymphatic system and returned back into our venous circulation. And we're going to discuss more on this at the end of our quarter when we discuss the lymphatic system. A bit more on fluid exchange across capillary walls. When we have a net filtration pressure, we're talking about the force responsible for moving the fluid across the capillary walls. And there are two forces that affect pressure, and that's going to be the hydrostatic pressure and osmotic pressure. What our hydrostatic pressure is a physical pressure of the blood flowing through the vessels or of fluid in the interstitial spaces. Our osmotic pressure is going to be the movement of solutes, whether it's plasma or tissue fluid, through a membrane, typically the plasma membrane, in the presence of a non-diffusible solute, such as a large protein. Large proteins aren't going to be able to move through the capillary walls easily, and the difference in protein concentrations between the blood and interstitial fluid is responsible for osmosis. If you remember, osmosis is the diffusion of water, and so the more solutes that we have in an area, the more the water is going to want to move to that area where we have a higher solute concentration. When we are standing, hydrostatic pressure caused by gravity increases blood pressure below the heart and decreases pressure above the heart. And what we can do is use our skeletal muscles to contract to help move this venous blood back up our veins to move it back to the heart. Our valves will also assist us to make sure that this blood does not move back down. Now let's discuss the control of blood flow in the tissues. We could do this through 3 different avenues. Our first one is local control, and in most tissues, our blood flow is going to be proportional to the metabolic needs of the tissues. Second, we'll have our nervous system that'll be responsible for routing our blood flow and maintaining our blood pressure. And lastly, hormonal control, where our sympathetic action potentials or nerve impulses are going to stimulate epinephrine and norepinephrine release. So when we have local control, we are going to call upon our precapillary sphincters to assist us. When we start to see that our tissues around these capillaries have a lower concentration of nutrients, such as oxygen, glucose, amino acids, or fatty acids, then our precapillary sphincters are going to relax and allow the blood to flow through in order to get those nutrients back to the tissue. Other things that will cause the precapillary sphincters to relax is if we have increases in concentration of vasodilator substances such as carbon dioxide, lactic acid, adenosine, adenosine monophosphate, adenosine diphosphate, nitric oxide, and potassium. And this as the pH is going to decrease. Now, if we have the opposite action take place and our tissues have a larger concentration of nutrients like oxygen, glucose, amino acids, and fatty acids, then our precapillary sphincters are going to contract. And when they contract, that closes up the lumen to our capillaries, and our blood flow is going to decrease to that tissue. Our sphincters here are also going to contract if we see that our tissue concentration of metabolic byproducts such as carbon dioxide, lactic acid, adenosine, and all of those other chemicals we discussed decrease. as the pH increases. When we talk about autoregulation of blood flow, we mean that we can maintain the blood flow by our tissues. We can change our arterial pressure over a wide range, but blood flow to our tissues are going to stay pretty constant. To illustrate this, our blood flow can increase about 7 to 8 times as a result of vasodilation of our metarterials and precapillary sphincters in response to an increased rate of metabolism. What allows us to have this autoregulation is vasomotion. And that's going to be the periodic contraction and relaxation of our precapillary sphincters. If we start to see changes within our tissue's metabolic requirements, then we can set up a long-term regulation of blood flow to those tissues. We can actually change the capillary's density over time. For instance, if you start exercising and we see that there is a greater metabolic need to that muscle tissue, we will increase the capillary density there in order to get it to receive more nutrients and oxygen. In order to best stay alive, we need to have a good nervous control of our blood flow in our tissues. So we are going to operate them based on reflex arcs, and they're going to involve our cardiovascular center of our medulla and our higher brain centers. In the medulla, in our cardiovascular center, we're going to have clusters of sympathetic neurons there. We break it down into a cardiac and vasomotor center, and this area of our medulla is going to be the excitatory part that is tonically active. So it's going to cause our vasomotor tone and also releases of norepinephrine. The inhibitory part can cause vasodilation by decreasing the sympathetic output. So notice how we are not talking about any role in parasympathetics here, but just the sympathetic. Our cardiovascular center is also going to receive inputs from our baroreceptors, our chemoreceptors, and higher brain centers. So what do those higher brain centers do for us? They're going to send reflexes that are going to regulate our blood pressure found in the medulla, and our hypothalamus, which is found right over here, and our cerebral cortex at the periphery of the brain. It can modify arterial pressure via relays to the medulla. Note that I've added new slides at the end of this PowerPoint, so please go over to Blackboard and you can update your PowerPoint with these last couple slides. The previous slides had incorrect information and were a little out of order. So we are going to get into our regulation of mean arterial pressure. And to do that, we have mechanisms in place to maintain this arterial blood pressure within our normal range values. Our mean arterial pressure, or MAP, is going to be slightly less than the average systolic and diastolic pressures because diastole lasts longer than systole. So our MAP is going to be 70 millimeters mercury at birth, 100 millimeters mercury from adolescence to middle age, and 110 millimeters mercury in healthy older individuals. And these are some equations that we have seen before. That MAP is going to equal our cardiac output multiplied by our peripheral resistance. Or we can also use the equation of heart rate multiplied by stroke volume, because that is the equation for cardiac output, and multiply that with peripheral resistance. Also note that MAP is going to be proportional to heart rate and stroke volume and peripheral resistance. So if any of these values increase, then our MAP is also going to increase. In order to regulate our pressure, we can use two systems, our short-term and our long-term. So we'll start off with our short-term regulation of blood. You may recognize the baroreceptor reflexes. These are the sensory receptors that are going to be sensitive to stretch. So the way that they can make changes to our blood pressure is by changing peripheral resistance, heart rate, and stroke volume. Remember that we find these sensory receptors in the walls of our large arteries of the neck and the thorax, the carotid sinus, the base of our internal carotid artery, and also the walls of our aortic arch. We're going to send action potentials that travel through the glossopharyngeal nerve, which, by the way, is a cranial nerve, specifically cranial nerve number 9. And that goes over to our cardio-regulatory and vasomotor centers in the medulla. From our aortic arch, we are sending information via the vagus nerve, which is also a cranial nerve, specifically cranial nerve number 10. And in these centers, we will have reflexes that will keep our blood pressure in homeostasis levels. So for instance, if we have normal blood pressure, then we are going to send low frequency action potentials through these nerves. But if we suddenly have an increase in blood pressure, then our vasomotor center is going to have the brain decrease its sympathetic stimulation of blood vessels, and our cardio-regulatory center is going to send increased impulses through our parasympathetic stimulation of the heart. At this point, our peripheral blood vessels are going to dilate, our heart rate will decrease, and blood pressure will decrease. Next, let's discuss the adrenal medullary mechanism. This is going to be activated by substantial increases in our sympathetic stimulation of our heart and blood vessels, such as a sudden large decrease in blood pressure, a substantial increase in exercise, or stressful conditions. So at this point, we are going to have increased stimulation. Let's say we're exercising, and that tells our centers in our medulla oblongata to send a signal down our spinal cord and out our sympathetic nerve fibers to our adrenal medulla in order to release epinephrine and norepinephrine. Our chemoreceptor reflexes are also going to be sensory receptors, and they're going to be sensitive to oxygen, carbon dioxide, and our pH levels of blood. These reflexes are going to be stimulated whenever we have decreases in our blood oxygen levels, increases in our carbon dioxide levels, or decreases in pH. Note that as our carbon dioxide levels increase, our pH levels are going to decrease. And the same thing is going to take place, but vice versa. So if we have our blood carbon dioxide levels decrease, then our blood pH is going to increase. So let's say we do have a decrease in oxygen, increase in carbon dioxide, and decrease in pH. Then we are going to have a decrease in the parasympathetic stimulation of the heart, which would cause an increase in heart rate. We would increase sympathetic stimulation of the heart, which also causes an increase in heart rate and also an increase in stroke volume. And we are going to increase sympathetic stimulation of blood vessels, which means we are going to have vasoconstriction take place. And lastly, we have our central nervous system ischemic response. This is going to take place when we have really low oxygen levels, increased carbon dioxide, or a decrease in pH in our medulla. At this point, our medulla is not getting enough blood flow, so it responds to help increase the blood pressure. And this will only take place in emergency circumstances where we see that the blood pressure is going to fall below the level of 50 millimeters mercury. At this point, our vasomotor center is going to be very strongly stimulated, causing an increase in blood flow to the brain by vasoconstriction. Now, this can't last too long. Remember, this is short term. So over time, the vasomotor center is going to become inactive. Now let's move into the long-term regulation of blood pressure. We have two systems or mechanisms in place here. We have the renin-angiotensin-aldosterone mechanism, and this is going to be something that you are going to be revisiting many, many times. So it's good to have this memorized pretty much right now. So let's start with when we have a decrease in blood pressure, our kidney is going to release a hormone called renin. Renin goes over and takes angiotensinogen that has been released by the liver and converts it over to angiotensin 1. From there, we are going to take the angiotensin converting enzyme in our lungs and capillaries, which will help convert angiotensin 1 into angiotensin 2. Angiotensin 2 will do a couple of things, but mainly for now, we will know that it's going to cause vasoconstriction, which will increase our blood pressure. And it will also release aldosterone from the adrenal cortex. Aldosterone is going to cause increased sodium reabsorption, which in turn will also increase water reabsorption into our blood, which will raise our blood pressure. Next, we have our vasopressin or ADH, antidiuretic hormone mechanism. So here we are going to have our blood pressure dropping, and we know that because our baroreceptors in the aortic arch, carotid sinus have told us this. At that point, the osmoreceptors are detecting an increase in osmotic pressure. And so our ADH is released from the posterior pituitary gland, which goes over to our blood vessels and causes them to vasoconstrict, which will lead to an increased blood volume and increased blood pressure. And the other thing is ADH will travel over to the tubules within your kidneys and tell it to reabsorb more water. When we reabsorb water into our blood, that's going to increase blood volume and blood pressure. These mechanisms can help regulate from minutes to hours to days or even weeks. This is a nice summary of the long-term control that takes place when blood pressure changes. So let's just go through an example here. So if we have an increase in blood pressure, what will happen is our renin-angiotensin aldosterone mechanism is going to detect that increase in blood pressure and produce angiotensin 2 and cause secretion of aldosterone from the adrenal cortex will decrease. The other mechanism that we haven't gotten to yet is the arterial natriuretic mechanism. Within the atrial walls, we have cardiac muscle cells that will detect an increase in atrial blood pressure and will start secreting this hormone. That will then cause vasodilation, decreases in the peripheral resistance to blood flow, and more sodium and water are going to be lost in the urine, and that results in decreasing blood volume, which in turn decreases the blood pressure. So let's talk about that hormone just a little bit more. This is an antagonist to aldosterone. Like we said, it's released from cardiac muscle when we have increased blood pressure and will cause us to have increased urinary production, which will reduce our blood volume and blood pressure. We can also have something called fluid shift take place, where we will have movement of a fluid from interstitial spaces in our tissues into the capillaries. And this is in response to a decreased blood pressure to maintain our blood volume and vice versa. Lastly, we have our stress relaxation response. Here we're going to adjust the blood vessel smooth muscle to respond to changes in our blood volume. So when blood volume is going to decrease all of a sudden, then our pressure drops and our smooth muscles are going to contract, and the vice versa will take place. So that is it for our chapter 21. If you have any questions, please feel free to reach out to me, and hopefully I will see you in our virtual tutoring session.